I'm pleased to introduce our next speaker, Professor Sarah Heilshorn. Sarah is a professor and associate chair in the Materials Science and Engineering Department and the director of the Gabel Laboratory for Advanced Materials. She holds courtesy faculty appointments in the departments of chemical engineering and bioengineering and is a Bass University fellow in undergraduate education. Her lab integrates materials science and protein engineering to design new bio-inspired materials for applications in regenerative medicine, organoid culture, and 3D bioprinting. Professor Heilshorn is a fervent supporter of diversifying the engineering community and serves in multiple leadership roles in order to help achieve this goal. She's a fellow of the American Institute for Medical and Biological Engineering and a fellow of the Royal Society of Chemistry. She currently serves as associate editor for the journal Science Advances, editor of the journal Acta Biomaterialia, and serves on the board of directors for the International Society of Biofabrication and the Tissue Engineering and Regenerative Medicine International Society. Welcome, Sarah.
Thank you. I am so excited to be here with you today and to highlight in particular the collaborative work that the Tsai Institute has enabled our group to do. I have a quick disclosure. And then I'd like to begin by asking you all to just take a moment and think, what makes you, you, right? What are your physical attributes? What are your cognitive attributes that are different? Do we have anyone in this room who's a twin? Oh, only one in a group this size. Statistically, there should be more. You, I'm sure, are different from your twin. And we call identical twins identical based on their genes, because their genes are nearly identical. And we just heard a beautiful story about how genes can change function. But the genes aren't the whole story, because in identical twins, their genes are nearly identical, and yet they're different. They are each unique. Although many of their physical attributes look the same, their fingerprints are different. Why are their fingerprints different? It has to do with the environment during development, the fluid forces that each are exposed to in the womb, the chemical gradients of the biochemicals that wash across. So you're a product of both your genes and your environment. And this is true for the cells of our nervous system as well. So this is a schematic, a very simplified, overly simplified schematic of neurons with glial cells and all of the other things surrounding the cells. To a material scientist, those look like materials. They're biopolymers. And they cause the cells to do different things, and different things with aging and health and disease. I'd like to point out this schematic was put together by a team of trainees that were specifically enabled by the big ideas. And so this is truly a remarkable team to be a part of that really spans departments from across the whole campus. And during the pandemic, when the trainees were unable to go in and do a lot of experiments, they wrote this really fascinating collaborative interdisciplinary review article. And so environment matters for trainees as well, right? Environment matters for ourselves, and it also means that we're producing trainees that think differently because they're part of this institute. And as partly evidence of that, Vivian, who was a graduate student at the time of writing this review, she's now a postdoc at MIT. She'll be returning next year as a new junior faculty member at Stanford. So let's think about the environment where we culture our cells when we're doing neurobiology studies, typically. These cells are often grown in a petri dish. It is a homogeneous liquid environment. It is a rigid plastic. It is two-dimensional. And we can learn amazing things from this culture system, but it's very challenging. Oh, I don't know why. You can't read, apparently. Some of the text shows up really light gray. That's a weird Google Drive file transport issue. I apologize for that. I'll try and read it for you, but I also have really bad eyesight. It says, how can we make personalized cell cultures that mimic real tissue, right? And that's what my group is really fascinated to do. How can we engineer tissues that begin to replicate, not fully, but in some ways, some of the properties of natural tissue? So all tissues are dynamic. They're constantly changing as we age. And it includes the living cells, but then also this non-living component of the biopolymers. And there has been decades of research that have shown ways that cells modulate these surrounding materials, and also that the materials can influence what the cells are doing. And so in my group, we're interested in leveraging fundamental polymer science that a materials engineer would learn to try to design materials for specific cellular needs and applications in neuroscience. And one of the strategies we like to use to accomplish this is protein engineering. And this is a little bit different from what many in the tissue engineering community do, where they either create their polymers using synthetic chemistry, or they isolate naturally evolved biopolymers, such as the alginate that comes from seaweed. Both approaches have really important advantages, but some also important limitations. In a synthetic material that a polymer chemist makes in the laboratory, it is really well defined. Great polymer chemists exist. They're much better chemists than me. They can make beautiful materials, but these materials typically are considered bioinert. They must be decorated with peptides or proteins to impart some biological activity to them. In contrast, materials that have naturally evolved are intrinsically bioactive, but we have very little control over them, and every batch tends to be a little bit different. So if we make our own recombinant proteins that we design the amino acid sequence ourselves, we hope to combine both sets of advantages into materials that are bioactive, but perfectly reproducible because we've designed them ourselves. Once we make these proteins, we formulate them into a hydrogel form factor. So gels are water-swollen networks, and they're mostly water, up to 99% water. This makes them very useful to create engineered tissue. We have good oxygen transport, good nutrient transport, and importantly, you can put a lot of cells in these materials because they're mostly water. The brain, for example, is nearly 90% by volume cells, okay? So you couldn't achieve that high level of cell density simply putting cells on rigid surfaces. We can also then design the amino acid sequence to hopefully mimic some of the native biochemistry that we're interested in studying that might change in different developmental processes or different disease processes. In my group, we use these protein engineered gels to do 4 key different types of applications and studies. And this serves as a roadmap for what I'm going to share with you today. We're going to start by talking about how these gels can be used to inject therapies, in particular a cell therapy. And then I'll tell a little story about using these materials to create cultures outside of a patient. And then if I have time, a very brief one about biofabrication. So spinal cord injury is devastating. We don't have regenerative therapies following a spinal cord injury. Our standard of care is mostly to prevent additional deterioration. But there's been many studies, both preclinical and clinical phase studies, showing that cell transplantation of many different cell types can help promote some regeneration of function and tissue structures. But, and the important part here is missing. This was in red in my file. It says, however, there is a delivery problem. And these data here hopefully highlight that. So you have a number of cells you want to deliver. You don't even get all those cells that you worked so hard to make, don't even make it into the injury site. And then within 10 minutes, another large fraction have passed away, and they continue to not survive over the coming months. So our engineering goal was, could we deliver a material that was designed to specifically keep these transplanted cells alive? And in particular, we started by doing an engineering analysis of why are the cells dying? And we identified 4 main challenges that happen over different time frames. And so we needed to design our gels to be able to have different properties across this range of timescales. Initially, many of the cells simply die due to extension, which rips apart the cell membrane as they're being delivered by these thin cannula into the injury site. Then, the central nervous system is under positive pressure. The cells simply reflux back out after you remove the needle from along the injection tract. The initial injury results in the loss of cells and the surrounding matrix, leaving big fluid-filled cavities. And so the cells have nowhere to sit onto. This can result in anoecus, which is a lack of matrix-induced cell death. And then long-term, that matrix needs to be there to physically support the movement of cells and their regeneration. Oh, and this moved over, so you can't see. Okay. So the cells, sorry. So the gel that we've designed, and it's hidden right here, sorry. There's a video of a syringe needle with a gel being pushed out. So the gel forms through assembly of peptide sequences. Peptides can come together and reversibly bind and unbind, which means this gel, when you apply a force to it, can flow through a syringe needle. When it comes out the tip, it immediately resolidifies and self-heals. And you can almost see a little bit of it. It's colored green. It's like tantalizing. It's teasing you. And this increases the number of successfully delivered cells over 500% by providing mechanical shielding to the cell membrane during the delivery process. And that was published work in a more recent unpublished, currently in peer review study. Here we're doing a rat mild cervical contusion at the C5 level model, and we are transplanting the cells. These are in a human pluripotent-derived motor neuron, and we're delivering them either in saline or in our gel, which we call shield, because it's meant to shield and protect the cells. And what we're observing is that after 6 weeks after the transplant, we can test whether our cells are still there and viable by staining for a marker that's specific for human cells. And we see that the number of cells at the site of transplant is fairly similar in our material versus in saline. But what is really different is the function of those transplanted cells. And that when delivered in our gel, that's this pink here, we see long neuritic projections that go up to 10 millimeters past the site of injection. So we're getting long elongation out into the surrounding endogenous tissues. And this then results in improvements, statistically significant improvements in the functional behavior of these animals. This is a mild injury, so it impacts one of the forelimbs and not the other, so we can use that as a control. And here I'm just showing 3 different types of behaviors that we're looking at. Sorry, I don't know why this, it's very interesting. So one is this exploratory behavior, and then we look at their strength. And then this is a model of their coordination as they visually look to see where the ladders are as they traverse this device. And when we compare across these metrics, we see that our cells delivered in our engineered gel are significantly improved compared to all of our other various controls. And this type of work was seeded through a seed grant from the Institute. Initially, this work started with 3 colleagues who have since moved on from Stanford, looking at transplantation of Schwann cells. We've now grown our team. The data that I've shown you was the work of a fabulous scientist in my group, Vanessa, and with new collaborations to look at the delivery of these human pluripotent cells so that we can begin to look at the integration of new circuits from these transplanted cells. What this says here that you maybe can't see is drug delivery to improve peripheral nerve regeneration and drug delivery to improve regeneration following stroke. So these are 2 other types of studies that are going on in the group right now where we're working collaboratively. Wherever you see a name underneath or a department title, these are my faculty collaborator colleagues, and names without departments listed are members of my research team. Okay. So next, so that was a story about designing materials to potentially transplant into a patient. And now we're going to talk about a slightly different project where we take samples from patients and want to grow them outside of the patient in vitro in the laboratory. And it combines these 2 topics together. So after me, you're going to hear from my collaborator, Sergio. We're really fortunate to have a real pioneer in neural organoids here on campus. And it's really impacted a lot of the studies that are going on in my own group. And so I apologize if this definition of organoid is not the current most up-to-date one. He can let you know if it is. So organoids are these collections of cells. And what's important is that they have these emergent properties where they can self-assemble and organize themselves if coaxed in the right way to create structures that recreate aspects of the actual tissue itself. And these have tremendous value in recapitulating features of the human brain. And in particular, what his lab has shown is that you can take organoids that are meant to replicate different regions of the brain, like the dorsal forebrain, the ventral forebrain, and put them together, and they will fuse over the time span of a couple days. And then these can be used to look at the different developmental processes, such as interneuron migration. Now, currently, when these assembloids are made, it is done through manual manipulation within a liquid cell culture medium. And as an engineer, that seems like a painstaking. As I was listening to Anne, I was also thinking like, oh my gosh, someone did all of that, right? It's a tremendous amount of work. So how can I, as an engineer, help to increase the throughput of these studies? And so the goal statement here is not showing up, is to design a technology that would achieve reproducible spatial patterning of these organoids. And so the system that my team came up with, they nicknamed it SPOT, for spatially patterned organoid transfer. We begin by having this little custom-made chip where each organoid is nestled, and we coat it with a temporary coating of a magnetic ink. That allows us to then come in with a bioprinter that has an electromagnet probe. We can turn the magnet on and lift up that organoid and then transfer it to a particular location. And we then turn the magnet off to keep that organoid there. And this pink is meant to represent what we call a viscoelastic media. So it's in between that of a liquid and of a solid. So it can keep the organoid floating there in three-dimensional space as we build up other organoids around it. Using this process, we can then pattern individual organoids into shapes like pyramids and rings and lines, and moving towards developmentally important and realistic structures. Once the fusion has occurred, we can then remove the fused assembloids and use them for studies of development and disease progression and track things such as migration across these interfaces. In one example, we're interested in using these to study migration of different types of brain cancers into these different brain-specific regions to look for patterns and potentially in the future be able to do drug screening by having multiple of these cultures made in a more reproducible, scalable way. So this project, as I mentioned, really was enabled by a collaboration with Sergio. The work on brain cancers include important collaborations with Van and Michelle. And I didn't include another collaboration that we have in the same space of looking at samples outside of the patient with both Kalina and Tony with new imaging modalities to look at brain tissue. So I have just a couple slides on this last topic. And as we heard from Anne, neural stem cells and neural progenitor cells, sometimes they don't scale up. They don't expand. We can't activate them. They stay quiescent. And it turns out this happens often when you take them and put them in culture. And so if you want to make a therapy for patients that uses these cells, we will need to make billions of them and need to make them very reproducibly. It's really hard to do that in two-dimensional culture. If you do it in three-dimensional culture, you can scale it down in terms of overall volume, but these cultures, neural stem cells tend to become fairly quiescent in three-dimensional culture. They simply don't grow well when you embed them within a gel. And so one of the goals in this project was to try to identify, are there properties we could engineer into these protein gels that would help to maintain these cells and get them to expand while maintaining their stemness. And in particular, we were interested in looking at the property of being able to remodel. So just as cells in our tissues are constantly remodeling the polymers around them, we wanted to incorporate that into our engineered materials so that we would have enzyme-responsive peptides that could respond to the enzymes that were being secreted by the neural progenitor cells. And this is really distinct from how most synthetic gels are made that are relatively static and don't change over time. And what we were able to demonstrate, so here we've created a family of gels where the mechanical properties are the exact same. All of the rest of the biochemical properties are the exact same. But we've modified the ability of these gels to be degraded in response to enzymes secreted by the neural progenitor cells. And we see that we have high cell viability in all cases, but we only get maintenance of these canonical neural stem cell markers in the three-dimensional gels that are remodelable. We also see increase in their beta-catenin signaling and hence their Ki67 markers of proliferation. And so this is a material that we can use to really scale up and create large amounts of activated neural progenitor cells for future clinical therapies. So this is just a short little summary of the walk and really focusing on, again, this idea that we can only do this work because of the environment that we're in, because of the Institute, and this very intercollaborative, interdisciplinary, and rather fearless group of individuals who are willing to talk with others who speak very different scientific languages. So with that, I'd like to thank all of my collaborators and also make sure to mention my sincere gratitude to the patient and patient families that have offered us to use their samples. Thank you.
Great. We'll take a few minutes for questions.
Thank you for a fascinating talk. I'm also an engineer, but I don't work with cells. I've been wondering, how do you get the magnetic ink off of the cells in spot?
Yeah, so the gel that we're using there currently is based on nanocellulose. So this is a plant-based biopolymer. And cellulose can be degraded by the enzyme cellulase. Because cellulose is not normally found in mammals, mammalian cells completely ignore cellulase, or at least on all of the control studies we've done so far, they don't seem to mind it at all. So when we treat with cellulase, it rinses all of the magnetic nanoparticles away, and then you're left with the assembloids. Yeah, thanks for the good question.
Thank you for the wonderful tour de force through how to apply some of these unique and really innovative biomaterials to neural systems. I was wondering, with your work on the stem cells and the materials and how you kept them in their stemness state, what you thought the most important kind of mechanical stresses on the cells would be to manipulate what state those stem cells are in.
Yeah, so in our mechanistic studies there, we found what was most important is that the cells be able to have physical contact with another cell. And it's the N-cadherin signaling, the cell-cell handshakes that are made, which then turns on additional beta-catenin signaling, which then turns on the transcription factors to keep those neural stem cell markers chugging inside the cell. And what's interesting is that it also seems to predispose the cells down different differentiation pathways later. And we've identified that certain gel formulas predispose these expanded neural stem cells to adopt more neuronal versus glial phenotypes when exposed to differentiation. So it seems that the environment that you use to expand up your cells also can alter what they're able to, how they enact different genetic pathways later to then differentiate into final cell products.
Because neurostem cells in humans are in quiescent states, are in hypoxic niches, have you looked at the effect of oxygenation on your cultures?
Yeah, that's a really fabulous question. We haven't for our neural cultures and neural organoids. In complementary work, we do a lot with intestinal organoids because intestinal stem cells can be isolated in a relatively minimally invasive way, shall we say, compared to neural stem cells. And there, we absolutely know that oxygen content matters tremendously. Absolutely.
Thank you.
Thank you very much, Sarah.